Are you Swiss and planning to move abroad, or have you already taken the leap? I'm really excited to tell you that SwissInfo has a new podcast coming out just for you. You can listen and watch it from the 25th of November. It's called Ade Merci Schweiz and is available in Swiss German and French. It covers everything you need to know about setting up your new life abroad. We speak to Swiss around the world who've already made the move, and we ask experts to share their experiences. You'll find Ade Merci Schweiz wherever you get your podcasts or in our Swi Plus app. Hello, I'm Jo Fay, and this is a SwissInfo podcast. SwissInfo is the multilingual and international public media company of Switzerland. Is there a drug that can prolong our lives? Scientists believe that we're getting close to finding that magic pill. In this episode, we're speaking to a molecular biologist in Basel about how a groundbreaking discovery he made 30 years ago is now fueling one of the biggest health trends of our time: longevity. Have you heard of the drug rapamycin? I hadn't until we made this episode. It's used to prevent organ transplant rejection or treat certain cancers and many other things. The drug is also known to have some impact on ageing. Our healthcare reporter, Jessica Davis-Plus, talked to the American-Swiss molecular biologist Michael Hall. He made some of the biggest discoveries about rapamycin. Here's Jessica.
Michael Hall is a virtual unknown in today's social media-inflated longevity scene. He hasn't published any books on why we age or been featured in any films on the secrets of centenarians. He doesn't have a side business selling supplements or red light therapy or other treatments found in many of today's longevity clinics. Hall's absence from the current longevity craze is all the more incredible given that his research is widely considered to have transformed our understanding of aging and potentially how to slow it down. In the early 1990s, he discovered a gene in yeast that regulates the growth and health of the cell in response to nutrients around it. He named it target of rapamycin, abbreviated to TOR, after the compound rapamycin, which was discovered from a bacterium. Study after study in animals shows that when you inhibit TOR, which was eventually named mTOR, you extend lifespan. There's now a rapidly growing community of longevity enthusiasts taking rapamycin. I actually only found out about Hall because he won the Balzan Prize for his work on the biological mechanisms of aging in 2024. I wondered, who is this person who seems to have done so much for the aging field, but whom I've never seen at any longevity events?
I can't believe you keep all of these things. This is amazing.
Yeah, this was in 2003.
I met Michael Hall where he works, at the University of Basel. He's now in his early 70s, yet still working past the official retirement age, which, here in Switzerland, is 65. I quickly learned that Hall encountered the aging field before his groundbreaking discoveries. In fact, it was even before he was a scientist.
My first interaction with science, with aging, was in the mid-1970s. I had a very, I would say, eccentric uncle, a self-made man, and later in life, one of his ambitions was to live to 100.
So this eccentric uncle, along with another of Hall's uncles and a friend, both medical doctors, decided they wanted to travel the world in search of what we today call blue zones. Those are regions in the world where it's claimed that people have exceptionally long lives.
The three of them went to Vilcabamba, that's in Ecuador. They went to Georgia in Russia. They went to, and they were going to all these places, on just to study. Mm-hmm. And then they're going to go to Okinawa, which is now recognized as the blue zone. These are all recognized as blue zones now.
Hall was still in his early 20s and between studies. He thought it sounded interesting, so asked if he could come along. His uncle said yes.
They brought this whole aging aspect to my attention. In the 1970s, nobody was talking about aging like today. It was so typical of my uncles to be so way ahead of the curve on this kind of stuff.
Do you remember learning anything about aging?
The conclusions were that aging is multifactorial. Mm-hmm. Genes, activity, diet, exactly the things I told you, which people are concluding right now.
It also turned out that some people had lied or been misinformed about their age. He still said the trip with his uncles made him appreciate aging as a scientific study. As he started to build his own scientific career, he remembers real scientists studying aging, but he also saw a lot of semi-science that made him skeptical.
Was there a reason that you didn't kind of dive, as you said, you had this indirect relationship with aging, but was there a reason that you didn't find yourself kind of
Well,
going
yeah.
more
My early experiences with this field was that there was just too much nonsense there.
What was an example of something crazy that was
I remember
happening?
people walking around in robes inside looking like Father Time.
Yeah.
People coming in off the street, with this, not even scientists. Everybody wanted to make a dollar too. Wanted to get some product. I mean, there were real scientists, but there was also just too much noise. And I was working on something else I thought was quite interesting too.
Hall did a postdoc in San Francisco where he was investigating how a protein gets transported into the nucleus of a cell. This provides critical insight into how a cell operates. But it wasn't until he moved to Basel when his work really took off. At the time, immunosuppressive drugs like cyclosporin, which is the active ingredient in Sandimune, and rapamycin had just been introduced. They were game changers for the medical field. They suppressed the immune system so that it didn't reject donated organs. In other words, they made organ transplants possible.
These drugs were extremely exciting, but nobody knew how Sandimune worked. Nobody knew how rapamycin worked. The immune system works by recognizing a foreign antigen, a foreign molecule, and it's not normal in your body, and does it by receptors on T cells. And so the antigen binds the receptor and activates the T cell. The T cells amplify, proliferate and amplify and mount an immune response. It was known that what these drugs did was they blocked these cell surface, these initial steps. The immune reaction were normal. Antigen, there was receptor was there, bound antigen, but everything downstream of that inside the cell, which created the amplification of the T cells, the pathway that signal into the nucleus to activate transcription, to make these functions. Nobody knew what that pathway was, but it was known that these drugs always blocked this. So something in the pathway from the cell surface, the receptor, to the nucleus. At that time, not a single signal transduction pathway was completely known. But what we knew is that the defect was somewhere in this intermediate step between the cell surface and the nucleus, and that's where, in very broad brushstrokes, where we were focusing our interest. How is information transmitted from this place in the cell to this? In our case, the information was a protein.
And so Hall went from researching nuclear protein localization to researching how these new drugs worked. And while most researchers did tests on mammal cells, Hall and his team took a different approach: yeast cells.
This is a radical idea because yeast cells are unicellular organisms. Why would you ever give a human drug to a yeast cell? Yeast like you make beer, wine, bread, and it was just physiologically completely irrelevant. But it goes back to the fact that in those days you couldn't do genetics with mammalian cells. CRISPR didn't exist, but you could do genetics with yeast, and we were a yeast lab, and we wanted to do genetics. And we also knew that rapamycin had been originally isolated from a soil sample collected in Rapa Nui, and had been originally isolated as an antifungal.
I should back up here and say a few words about rapamycin. It was discovered in the 1960s from a soil sample collected by Canadian scientists that traveled to Easter Island, which is also called Rapa Nui, hence the name rapamycin. It wasn't actually called that until 1975 when scientists identified it as an antifungal agent. It's because of these antifungal properties that Hall knew it could be studied in yeast. The fact that Hall was in Basel, close to Novartis, which at the time was separate companies, Ciba Geigy and Sandoz, also meant he had access to people working on these drugs.
It's just a matter of being in the right place at the right time. That's the beauty of Basel. So, this is a town where there's so much science that you bump into these things.
Okay. Do you remember the aha moment when you...
Yeah, I'm asked this question a lot. I think this aha moment is is a little exaggerated.
Okay,
I don't
it's like
think,
years.
Yeah, I say it's a series of mini ahas. I mean, when we first saw that rapamycin did something to a yeast cell,
Yeah.
maybe our approach is going to work. When we cloned the genes, and everything made sense. The mutations blocked the binding of rapamycin to TOR, and all this. Everything fell into a beautiful, very logical, very irrefutable story. But this is not a specific, precise moment where you pick up something and think,
Hall's first major finding was published in 1991 in the medical journal Science. He identified two genes, TOR1 and TOR2, that when mutated were resistant to rapamycin's effects on the cells. His team sequenced the gene to identify the protein encoded by it. Further research found that TOR is a central controller of cell growth, something Hall describes as one of his most gratifying discoveries.
people think our discovery of TOR is our major discovery, but I don't think that's our major discovery.
Okay.
Our major discovery came later, and that was the fact that cell growth is controlled.
That cell growth can be controlled, and that's probably what influences.
Look at the thing of this: there are three basic things a cell does: divide, grow, and die. People had focused on the division part, cell cycle control. That was well known. Increase in cell number. The death part, that was already well figured out. But the growth part was ignored. And the reason that this was completely ignored was the fact that they thought there was no active mechanism controlling cell growth. In other words, it was a passive process where if you fed nutrients to a cell, you gave them building blocks, and the cell took the building blocks and grew. We had to convince people that growth is controlled and TOR is the controller. And today, they would think you're crazy if you said cell growth is not controlled. I mean, it's incredibly naive. Looking back, it seemed incredibly naive to think
To even question
that cell growth is not controlled.
Yeah.
It's such a fundamental thing, And now we have all these diseases based on aberrant cell growth, like cancer.
Hall started publishing on these findings in 1996. This new knowledge about TOR controlling cell growth revolutionized cancer treatment. Pharmaceutical companies went on to develop a new class of cancer drugs known as mTOR blockers. But back to aging. Hall also realized something else. TOR responds to nutrients.
Another major discovery we did was to discover that TOR responds to nutrients. That's very simple. It's what senses nutrients and tells the cell to grow. It's very simple terms. TOR is what makes us grow and we eat. So we have TOR that connects nutrients, growth. If you put the dots together, you say TOR could also control aging then.
When TOR is suppressed by taking a drug like rapamycin or by fasting, it shifts the body from a growth mode to a conservation and repair mode. This activates autophagy, which cleans out damaged cells. Without autophagy, damaged cells accumulate, which can lead to age-related conditions such as osteoarthritis and neurodegeneration. This explains why fasting or restricting calories is frequently cited as one of the ways to slow down aging. Rapamycin works in a similar way to eating less calories, and this was only known thanks to Hall's findings. The first real validation of Hall's findings in the aging field came out a few years later. In 2003, a scientist from the University of Fribourg in Switzerland found that blocking TOR in worms increased their lifespan by 20 to 30%. Numerous studies have been done on other animal species since then.
Then it was shown a couple of years later in flies, worms, yeast, and flies. And then in 2009, there's a huge impact paper. It was shown that rapamycin extends lifespan of mice.
Mice, okay.
And this was the first pharmacological extension of lifespan in a mammal. This was really explosive.
There is now a major trial to see if a low-dose rapamycin treatment can extend the lifespan of older dogs. Even as more results emerged, Hall never worked on TOR and aging directly. He told me he thought he had bigger fish to fry. Hall's respect for the study of aging has grown, and while it is still filled with hype, Hall said there is much more rigorous science behind it than in the past. Today, Hall's findings are often mentioned in aging conferences, and he's often praised for his discoveries in longevity circles. But it's now 2025, and we still don't really know what rapamycin does in humans. I asked Hall about this.
Well, we can do a trial with humans. You can't do an experiment with humans. There are a lot of people. There's a whole online community which is taking rapamycin.
So, but why is it so hard to, I mean, we do clinical trials of many...
Because aging is not a disease.
Because aging is not...
You can't do a clinical trial on something that's not a disease.
Rapamycin is being tested in low doses in humans against certain age-related diseases like Alzheimer's. Even without the scientific evidence, hundreds of people are said to be taking rapamycin off-label for aging reasons, including medical influencers like Peter Attia, who wrote the book Outlive. Some claim to have seen mild benefits ranging from weight loss to pain relief, but some have also reported worrying side effects. Brian Johnson, who is the founder of the Don't Die movement and is known for his documentary on Netflix, decided to stop taking rapamycin after 5 years of experimentation. He said that the drug had too many side effects, including soft tissue infections and an increased resting heart rate. Still, rapamycin today is the only drug that has consistently extended the lifespan of different species. Michael Hall, who doesn't take rapamycin, says that while it may not ultimately be the magic pill, any drug to extend our lifespan will inevitably lead back to TOR.
The aging people saying, let's stop treating these individual diseases, creating different, different silos and different budgets. Why don't we just target the aging process? In one fell swoop, we will solve all the aging-related diseases, which I think is probably right. I mean,
You're quite hesitant about really...
What you'll be working on is TOR. I mean, TOR is sort of at the center of all this. It's involved in all these diseases, and this is where the knowledge of TOR is so important, I think.
Very interesting stuff. Thank you for bringing us that report, Jessica. Just how plausible do you think it is that one day we'll get just one pill that targets a whole range of different problems associated with getting older? Do you think that's something that could happen?
I'm not sure if it'll be a single pill, but I do think that there is progress in this area of looking at the root cause of some of these diseases as opposed to tackling each disease individually.
Okay, thank you very much, Jessica. Over the previous episodes in this season, we talked a lot about technical innovations and new kinds of drugs for old age. But quality of life in old age is also about psychology and perception. We'll talk about this with ageing expert and psychologist Dr. Christina Rucker. That's coming up in the next episode of the Swiss Connection Science Podcast. Today's episode was recorded and edited by our science and video journalist Michele Andina. For more content, visit our website, swissinfo.ch. I'm Jo Fay. Thanks for listening.